In corn, the grain-field period is divided into 6 stages. The first one is silking stage or growth stage R1 is when the silk emerges, and this is technically recognized as the first stage of the reproductive period. The second stage is kernel blister stage or growth stage R2. This is about 10 or 14 days after silking and is when the kernel become whitish on the cob and contain abundant clear fluid. The third stage is kernel milk stage or R3. This is about 20 days after silking. This is when the kernels are mostly yellow and contain milky white fluid. The fourth stage is kernel dough stage or R4. This is about 25 days after silking. The kernel's milky inner fluid is changing to a pasty consistency. As starch accumulation continues in the endosperm. The fifth stage is kernel dent stage or R5. This is about 40 days after silking. All or nearly all of the kernels are denting near their crowns. And the final stage is physiological maturity. This is about 55 to 65 days after silking. Kernel dry weight usually reaches its maximum and kernels are said to be physiologically mature and safe from must. The pattern of maize kernel weight responds to changes in assimilate availability per kernel during the grain filling period differ from the pattern obtained for other grain crop species like wheat and soybean. Decreases in the post-flowering sourcing ratio promote large reductions in final kernel weight, while increasing the ratio had minimum effects in increasing it. This suggests that maize plant set an individual kernel sink potential early in grain filling that places further kernel growth close to a saturated assimilate availability condition for biomass accumulation. Based on this, the hypothesis is that plant growth rate per kernel set around flowering could help estimate source availability per kernel at early grain filling stages in a simple way. This estimate of source availability per kernel should explain differences in final kernel weight promoted by genotypic or environmental differences. If it is true that maize kernel usually grow near saturated assimilate availability condition during effective grain filling period, in turn, kernel growth rate during the effective grain filling period is the main maize kernel weight determinant and depends on the zinc capacity established early in development. Therefore, if kernel weight differences among genotypes or environments are related to the source availability per kernel prior to the beginning of active kernel growth, they should be explained by the kernel growth rate during the effective grain filling period.
Methodology. Well, we can see first that experiments were realized into different experimental fields. The first one was the experimental field Department of Plant, and the second one was experimental field of Nideris, Argentina. There are a few important things that we have to know. The first one is that the soil were silty clay loam type. Another one is that they used 12 different maize hybrids and they were commercial, almost all of them. Both experiments treatments were applied at the same distance and at the same day. Shoot biomass was determined after drying plants in an air force oven at 65 degrees for at least 1 week. In each experiment, 30 plants were selected randomly 15 days before male flowering. Beginning on 7 days after silking, the apical ear sheet of one plant per replicate was harvested every 4 or 6 days. Dry weights were determined after drying the kernels in an air force oven at 70 degrees for at least 96 hours. Otherwise, stem volume was calculated from plant height and the stem diameter at the base of the stalk. The density that they applied was 90,000 per hectare. Then they applied nitrogen 100 kilograms per hectare. Well, to finalize the methodology, in the next table we can observe some of the different genotypes and some characteristics of each one, like for example the PGR around flowering, or the KW or kernel growth rate, and the most important part is the total grain filling duration, because that's our results.
Results. Genotypes different in PGR around flowering, as you can see in the table, and these differences were very consistent in both environments in spite of the significant side effects. Mean values of PGR around flowering were lower at VM than at VT, and a range between 171 and 360 milligrams for the former and between 251 and 352 milligrams. The latter genotypes also different in number of the kernels set per plant, but a genotype X site interaction was detected for this trait. Genotypes show no prophylaxis, so kernel numbers were always from the uppermost ear. Average across genotypes and environmentals, the range explored for the trait varied between 469 and 613 kernels plant. To this part, we need to look at the table. Look the variation in PGR around flowering and in the number of kernels set per plant produce a wide range of PGRs per kernel around this period, leading a genotype and site. Significant difference. Values of PGR per kernel around flowering varied from, as you can see here in the table. When data from all these states, genotypes and sites were pooled together, increasing PGR around flowering always led to an increase in PGR per kernel. As expected from Figure 1, as you can see, and as a result from the curvilinear response between kernel number per plant and PGR vacuum flower when PGR values are higher than 140 to 150 milligrams. During the effective grain filling period, PGR per kernel also showed significant genotype difference, although a significant genotype X site interaction was found. The range explored of PGR per kernel during the effective grain fill period was between 0.12 and 0.25 milligrams across all genotypes X site combination. Thus, genotype and sites help explore a wide range of PGR per kernel during the both early and late grain filling. Average across site final KW from spilate for position 10 and 15 from the bottom of a apical arm show a wide range among the 10 genotypes in the table 1, which varied from 281 to 360 milligrams kernel. Genotypes different in both kernel growth rate during the effective grain field period and total duration of grain field. Difference in kernel growth rate during the linear phase of kernel growth were strongly related to PGR per kernel around flowering. This was no relationship between kernel growth rate during the effective grain filling period and PGR per kernel during this stage. This indicates plant growth per kernel during the lag page, and not from the life phase onwards, was the promoter of the kernel growth, the variation observed among genotypes and environments. Nevertheless, although differences in KW were mainly related to change in kernel growth rate, genotypic difference in grain filling during duration were evident. This genotypic difference in grain filling duration determined that condition early in grain filling represent as PGR per kernel around flowering, help explain 66% of the variation in kernel growth rate, but only 40% of the total variation in KW. Genotypic difference in total duration of grain filling were related to variation in the ratio between VGR per kernel during the effective grain filling period and kernel growth rate during the same stage, as we can see here in the figure 4. As both rates varied across hybrids and sites, we hypothesized that the relation between them would serve as a good estimate of the source-link ratio during the last part of grain filling. A trend was detected for longer grain filling duration under higher sourcing regions during the effective grain filling periods, which helped explain the observed negative correlation between kernel growth and rate and grain filling duration. This trend, however, had minor effects on the kernel weight at physiological maturity, as the genotypic variation observed from the radio was very large and was not matched by a comparable variation in grain filling duration.
This image shows the phenological stages of the plant, vegetative and reproductive phase. Our article explains that the maize plants establish an individual potential kernel sink during the early kernel development. This cites the further growth of the kernel close to saturation condition that affects the assimilation for the biomass accumulation in the grain. There are different factors that determine the relation sourcing. The differences in the kernel growth are related with the early capacity of kernel sink. These factors play an important role in plant growth rate and the final kernel weight. The first one is the density. The second is pollination treatment. Pollination treatment affects the number of kernels for the same plant growing condition, so the same amount of assimilate produced around the flowering is distributed over a different kernel number. And the last is stress caused by drought. Our attempt to place 12 genotypes together has drawn attention in the existing wide variability in the plant growth rate around flowering, leading to similar kernel numbers, giving light to important genotype difference in biomass partitioning to the ear or in reproductive efficiency in kernel set. Kernel filling is related to the capacity of its genotype to keep a high production of biomass after the stage of flowering. The theoretical frame proposed for the determination of genotype and environmental difference in maize kernel sink potential. Environmental conditions affect kernel set through change in plant growth rate around flowering. This relationship affects the plant growth rate per kernel early their development, making kernel establish a differential kernel sink potential. Genotypic difference in the relation between kernel number per plant and plant growth rate around flowering will impact the resource availability per kernel and the kernel sink potential. For simplification, only show the numbers of kernel set per plant from the apical ear, but appearance of secondary ears at large plant growth rates will impact resource availability per kernel and the kernel sink potential.
The paper was not so well structured. It was a bit difficult to understand, but in the end we were able to extract the information from it, and now very important data that help us to understand the importance of source-sink relationship.